want to start with a few announcements. One is that if you are interested in a wellness unit, or maybe need a wellness unit, or just interested in working out in Bath campus, there is a course that meets on Saturdays to help maintain the trails in Bath campus, removing a invasive species that Brent Smith hates. Anyway, that is available. It wasn't posted on, during the registration thing, so, but it is, I think, available now for those of you who are interested in working in Bath campus or want or need a wellness credit. Other announcements include the quiz. No, the problem set that is due on Friday. Remember that. That's due on Moodle on Friday. And its goal is to help you remember and learn physiology related to the endocrine system. The quiz, that is again, you pick it up at CSD desk, take it and turn it in there. It's on paper. That will cover anatomy mostly. So for the endocrine section, the anatomy will be matching and fill in the blank. And for the leg anatomy section, that'll be a labeling exercise. So your tools for the first part of the quiz, the endocrine part of the quiz, are materials from this class, from classes and from the key concepts lists. And your tools for studying for the leg anatomy section are the coloring book. And I want to point out that on page 66 of the coloring book, which is not an assigned page, but it's a review page for the leg muscles. So that will be helpful for studying leg muscles. You can use that for self-testing, which is a useful way to learn. So that's the quiz. In lab this week, Thursday night and Friday morning, we will be using the cadaver to review for the practical quiz, which is next week. So there will be, ways to work with your teammates on the cadaver to help prepare, help solidify your understanding and knowledge for the practical quiz. That's pretty much all the announcements I wanted to make. Oh, I did post the answer key to exam 2. It is up on Moodle now. There's a chance I will have to take it down. So if you want to make sure you have, for at least for some period of time, so if you want to make sure you have access to the answer key, you want to download it, just in case when you want it, it's not available on you. Okay, so now we're going to turn our attention pretty much where we left off on Monday, talking about the endocrine response to physiological and psychological stress. Here's a slide that I showed on Monday. So the idea is that our brain detects stress, physiological and psychological, and our hypothalamus processes that stress. The hypothalamus communicates directly to the posterior pituitary, and it communicates indirectly through CRH released into the portal circulation at the median eminence to the anterior pituitary. Anterior pituitary corticotropes, specialized cells in the anterior pituitary that have receptors for CRH and release ACTH. Those are the corticotropes. Release ACTH in the systemic circulation, and the target of ACTH, that is to say where there is high expression of ACTH, is in the cortex of the adrenal gland, which is stimulated to produce and release cortisol. Cortisol has multiple effects. It is required for homeostasis, for just basic body homeostasis. And in addition to simply maintaining metabolism, it increases blood glucose. It is a growth inhibitor, so it maintains homeostasis, but that means it's not allowing proliferation or growth. And it is an immune inhibitor. So cortisol is an immune suppressant. Not sure I don't know enough about the immune system to understand why inhibiting the immune system would be adaptive for a stress response, but it is, or at least it's there. So you may have heard of drugs like prednisone that are glucocorticoids. They're analogs of cortisol that are used to assist in healing and, well, mostly to inhibit immune inflammation, immune-dependent inflammation. So there are steroids, drugs, that are anti-inflammatory by inhibiting the cells that release inflammatory molecules. This system is called the HPA axis. So if some physiologist or endocrinologist or endocrine physiologist refers to HPA, this is what they're referring to. Hypothalamic pituitary adrenal. So let's expand on this a little bit, expand on stress. Now this slide is, in some ways it's frustrating because some of the material on this slide depends on some of the physiological knowledge needed to understand the full depth of this slide depend on AMP2. And so this is where there's a little bit of a disconnect, or, the fact that we have two separate courses makes it a little bit complicated. So for those of you who've taken AMP2, I'm going to give you keywords that I hope will trigger memory of things that we discussed in AMP2. For those of you in this class, I'm not going to hold you responsible for AMP2 material. So it's just sort of a appetizer for AMP2 if you choose to take AMP2. So let's start with an easy one. Physiological stress is usually associated with an increase in blood pressure. So this is not easy, but that's where we're going. It's an increase in blood pressure. Maintaining blood flow to the key organs like the brain and the muscle in order to respond to that physiological stress. Blood pressure is where we're going. What we're going to go over here, the blood pressure. Stress, detected by the brain, causes the hypothalamus, and the direct interaction between the hypothalamus and the posterior pituitary. So remember, hypothalamic neurons, some of the hypothalamic neurons have their release site in the posterior pituitary. So this arrow is from the hypothalamus, but does not depend on CRH. CRH is going to the anterior pituitary. Okay, just making that clear. So CRH does not do this. The hypothalamus, these are axons from the hypothalamus. This is CRH into the portal circulation. So maybe that's the way to label this. Is label this arrow axons and label this arrow portal circulation to make it clear that this is not due to CRH, but this is. Is that clear? This slide, I built this flow chart and it's got some funny things in it. So, hypothalamus integrates this stress, causes the posterior pituitary, some of the cells in the posterior pituitary, some of the hypothalamic neurosecretory cells that release their hormone in this posterior pituitary, some of those release vasopressin. And vasopressin, those of you who took AMP2 should be thinking, okay, yeah, vasopressin, cortical collecting tubule, GS story, aquaporin, increased water reabsorption, increased blood volume because of water reabsorption, increased blood pressure because of increased blood volume. So everybody, I want you to know this, but I'm trying to key into those students who've taken AMP2 to remember some of the physiology that's associated with this hormone. Okay, blood pressure. Here's another response to stress. Other hypothalamic neurons release CRH in the median eminence. Cortical circulation triggers the anterior pituitary to release ACTH. This is what we saw in the previous slide. ACTH binds to receptors in adrenal cortical cells, cortex cells of the adrenal, and they release cortisol. Cortisol has multiple effects: anti-inflammatory, which is now written out explicitly, anti-immune effects, decreasing in prostaglandins, which is you don't have to worry about that. Just think of multiple effects of cortisol. In addition, cortisol augments the effects of glucagon and inhibits the effects of insulin. So I'm going to just put that out there now, and we're going to talk about glucagon and insulin in a few minutes. So let me just put that out there, that cortisol augments glucagon effects and inhibits insulin effects. That is, by doing that, it stimulates glucose release from the liver. So what this is in addition to increasing blood pressure, we're increasing blood glucose. Because the idea is in stress, the muscles need an energy source, the brain needs an energy source. One of our richest, sort of fastest acting energy sources is blood sugar. It also increases the breakdown of fats, but this is slower. This is faster. Stress is all questions about that. Stress is also detected by the sympathetic nervous system. So this is not part of the hypothalamus. This is separate. But the sympathetic nervous system, you know from the previous section of the course, releases epinephrine and norepinephrine. Epinephrine and norepinephrine have multiple effects. Now again, I'm going to AMP2. Those of you who took AMP2 should be thinking, oh yeah, epinephrine, beta-adrenergic receptors, GS, heart rate, increasing cardiac output, alpha-adrenergic receptors causing vasoconstriction, increasing cardiac output and increasing constriction, systemic vascular resistance, increasing blood pressure, right? So, again, I won't test you on that business, that incredibly important, very abbreviated story about how epinephrine and norepinephrine increase blood pressure here. That's AMP2. Those of you who have taken AMP2, I hope that it's ringing some bells. Those of you who didn't and are interested in how the sympathetic nervous system changes our heart rate and our blood pressure and our respiratory rate and the effects on glucagon and insulin, take AMP2. So let's continue this. So there is a pretty complete flow chart of the stress response, including the HPA axis right there, but not limited to the HPA axis. Let's do this exercise together. Okay, so this is the HPA axis. What I want you, I gave this to you as a blank. So over here, I'm asking you to put in the names of hormones. I've given you this one. So in these boxes, put in the names of hormones that are on this table or on this flow chart. In this box, I want you to put the anatomical location where this hormone is released. And in this box, I want you to tell me the cell type and receptor that responds, the cell type in the anterior pituitary that responds to the hormone that is released here. Okay? So I just want to flesh this out. Hormones over here, anatomical location here, and the cell type and receptor in the anterior pituitary that, that responds to this hormone and releases this hormone. So go ahead and let's take 1 minute for you to fill that in. And, oh, and then, you know, what are some of the main effects of this hormone? While we're working, online students, is this, I'm doing a sound check now, I should have done this earlier. Everything okay sound-wise?
Yeah, I can hear you clear.
Good, okay, thank you. Okay, let's start filling in the boxes. What's the name of this hormone released by the hypothalamus in the HPA axis? I heard it. Somebody say it loud. CRH. Corticotropin releasing hormone. What's the name of the hormone released by the anterior pituitary cells that respond to CRH? ACTH. Just as a way to make this make a little bit more sense, another name for ACTH is corticotropin, right? So this is corticotropin releasing hormone, and this is corticotropin, but it's usually called ACTH. So the name here is, they're not very helpful. It's helpful if you remember corticotropin is ACTH. All right, what is the name of the anatomical location at which CRH is released by the hypothalamic neurosecretory cells? Median eminence. You could also call it the infundibulum, that's an anatomical name. You could also say portal circulation. Those would all be correct answers. Okay, what's the name of the cell type that responds to CRH and releases ACTH? The name of that cell type in the anterior pituitary. somebody online, I'll look at you. I'm looking at you online. Somebody online yell out the name of that box, for that box. You thought you could just sit there and listen, huh?
Is it a Corticotroph?
Yeah, it is, corticotrophs. What's the name of the receptor that corticotrophs express? This is really, I mean, this is so straightforward that you're going to wonder if it's a trick. It's not a trick. It's the CRH receptor. Okay, and now let's fill in the box at the bottom. What are some of the effects? Well, let me just give you the answer there, because, you know. What are some of the main targets? Well, the targets are right here. Liver cells, fat cells, muscle cells, immune cells. Those are the target cells. That's given to you. Some of the effects are to increase blood glucose by increasing release of glucose from the liver, by increasing conversion of fat into blood glucose on fat cells. Muscle cells also contain glycogen, so they can release glucose. Immune cells, it's what we're going to inhibition of, let's just say, inflammation. I don't remember what I have on the answer key, which is the next slide. So your slide should look something like this. Let's fix this. So now your handout should look something like this. And this answer will be on Moodle at the end of class. Immune suppression, anti-inflammatory, metabolism of fats and glucose into fats and protein into glucose, synthesis of glucose and release of glucose from glycogen, blood glucose. Okay, so now, let's remember, this is a map of where ACTH comes from. What are the cells that release ACTH? The corticotropes of the anterior pituitary. What stimulates them to release ACTH? CRH. What is the story here? The story here is ACTH is proteolytically cut from a precursor. The precursor has a name, pro-opiomelanocortin. So now what I'm asking you to do in this slide is to write out what else we get, or what do we get when we proteolyze POMC. So obviously, one of the ones that we're talking about here is ACTH. That's the main story. But I want you to fill in this blank with the other products of POMC cutting. What are they? The answer's on the screen. So somebody tell me another product of POMC cleavage. LPH. Okay, so now we're off and running. What else? MSH. What else? What does this one stand for?
Beta endorphin?
Okay, well, that's this one, beta endorphin, so endorphins. Several endorphins, beta endorphin, well, at least one endorphin, but I think there's more than one. And what else? What is this one right here? Forget the net, what is anchor? And kephalin. These are the endogenous opioids that interact to reduce pain, right? So we have endogenous painkillers that are released in stress. So the answer is here on this slide. ACTH, MSH, and opioid peptides. I didn't even include LPH, but it's the correct answer as well. So let's use a problem, a clinical problem. Not so much, I'm not going to test you on clinical issues, but I think understanding clinical problems can help you understand the basic physiology that I can test you on. So there is a disease called Addison's disease, which was discovered by some guy, or at least characterized by some guy named Addison. It's primary adrenal insufficiency, or primary hypocortisolism. By primary, it's referring to the fact that the defect is in the adrenal gland, not upstream. It's the defect is in the cells that release cortisol. Hypo means low, so hypocortisolism, low cortisol, primary means because the adrenal is not releasing enough cortisol. Some defect in the synthetic pathway or the response, the receptor expression of the adrenal cortex, so it's not releasing the required amount of cortisol. And so cortisol is important, and so you can imagine that low cortisol is associated with, sort of general malaise, general poor health, fatigue, muscle weakness, weight loss. I'm not going to test you on that, but it's all true. But here's an interesting one. We're going to take a minute or two. I want you to explain why primary hypocortisolism would lead to hyperpigmentation of skin. People with Addison's disease develop darkening, especially in skin that is, white people, pretty pale. Hard to detect on people who, have dark skin naturally, so that's a hard thing to detect because primary hypocortisolism can happen in black people too, and this is hard to see. But in white people and in Asian people, there is a symptom, a sign, hyperpigmentation. Take a minute, try to figure out why the problem in the adrenal gland would lead to hyperpigmentation of skin. And I'll give you a hint. The answer is in this slide and this slide. So if you integrate your information and your knowledge about this slide and this slide, you will understand why there could be hyperpigmentation. Let's take a minute here and work on that problem. So, the answer could be, a perfectly correct answer could be written or spoken in four or five words. Doesn't have to be, obviously you can elaborate more than that, but you could come up with a reasonable answer to this question in 4 or 5 words. And here's the question. Anybody want to suggest an answer? This is a problem. This is not easy. This is not memorization. This is figuring it out. So this is the sort of thing that I could include on an exam. Anybody want to venture a guess? Sarah?
Well, since it's high-low, I would assume that the cortisol levels are low, so then there's going to be a negative feedback loop to produce more cortisol. And then doing that, like more CRH, more ACTH, which means that there's going to be more POMC processing, which also makes an increase in MSH, which leads to more melanocytes.
Perfect. So I'll say it louder so that everybody can hear it. The reason is low cortisol, low negative feedback. You didn't quite say that. You said negative feedback, but it's really loss of negative feedback. Low negative feedback, more positive feed forward due to loss of negative feedback. There's still this negative feedback, but it's not the full level of negative feedback. So loss of negative feedback, that's what this pale red is supposed to show. Loss of negative feedback due to low cortisol. Excess feed forward. Excess CRH. Excess stimulation of the corticotropes to produce ACTH. The way ACTH is produced is by excess expression of POMC, making this product, but also making this product. Excess production of melanocyte stimulating hormone. Melanocytes are the cells that give skin color. And so excess, so here's the answer. Low cortisol, low negative feedback, excess CRH from the hypothalamus, excess stimulation of cortisol, excess production of POMC, and therefore excess production of MSH, stimulation and proliferation of the melanocytes that give skin color. So this is the type of question I could ask on an exam to get you to integrate and solve problems with the information that you know. There is, I just want to make it clear that the causes of primary hypocortisolism of Addison's disease are multiple. It could be autoimmune, the immune system attacking the cortical cells, the adrenal cells. Infection can cause the immune system to attack the adrenal cortex. There are genetic defects in the adrenal cortex, so there's multiple effects. But if it's multiple causes, but if it's primary hypocortisolism, we lump that into Addison's disease. There is such a thing as secondary hypocortisolism. Secondary hypocortisolism refers to things upstream. So a defect in the hypothalamus or a defect in the corticotropes would lead to loss of positive feed forward, and that would also cause hypocortisolism. But it would not trigger hyperpigmentation because, it could, it depends. But the secondary hypocortisolism is a defect in the positive feed forward, not in the cells that produce cortisol. So that's secondary. There is another disease that I'm not going to spend a lot of time on. It's called hypercortisolism, Cushing's syndrome. So a doctor named Cushing characterized it. It's got a bunch of very strange symptoms. They're very characteristic. And it's often due to a tumor in the pituitary, that is to say, excess growth of pituitary corticotropes, too many cells, too much production of ACTH, too much stimulation of the cortex to make cortisol, or it can be due to a tumor in the adrenal. Too big an adrenal gland that is too big, cell proliferation, too many cells responding to normal amounts of ACTH, producing too much cortisol.
Bob,
Yeah.
why would Cushing syndrome cause only upper body obesity when we find that hormones travel through the bloodstream throughout the whole body?
I have no idea.
That's fair.
I have no idea. But it is characteristic from the shoulders up. The face gets fatty and swollen, moon face. Fat deposits on the shoulder. I don't know why those parts of the body respond to excess cortisol more than other parts of the body. I'm not sure anybody knows, but it's a characteristic sign of Cushing syndrome.
Gotcha. Thanks, Bob.
All right, so now let's turn our attention to the hormones that control blood glucose and growth and metabolism. So that's where we're going to go, and then we'll take a break. So the hormones we're going to talk about first are insulin and glucagon, and then the hormones we're going to talk about are thyroid and growth hormone. Okay, so first we're going to talk about insulin and glucagon. What makes them, what stimulates them to be made, what are their targets, and how does it work at the target? So insulin, everybody's heard of insulin. Let's unpack your knowledge about insulin. It's a peptide hormone. The stimulus for the release of insulin is high blood glucose. So in someone who is typical, a healthy, typical person, we're not talking about diabetes, the stimulation is having eaten a meal, putting food in your body, your intestines. Now I'm talking to the A&P2 students, remembering, okay, now think about all that digestive anatomy and physiology you learned about. All those, all that technique of taking a meal, protein, fat, carbohydrate, turning it into nutrients that are released into the blood. Some of those nutrients are straight up blood sugar, glucose, from carbohydrate metabolism, carbohydrate digestion. What am I asking you to remember? Oh yeah, amylase in the saliva, amylase in the pancreas, breaking down of carbohydrates, then sugar, glucose into lactose, from lactose uptake in the intestines. Remember all that stuff? At least those words should, again, I'm talking to the AMP2 students, or I'm giving those of you who are scheduled or planning to take AMP2 a preview. You eat a meal, blood glucose goes up, it stimulates insulin to be released by the beta cells of the exocrine pancreas. So the pancreas, I'm sorry, the endocrine, we're in endocrine, by the endocrine pancreas. The beta cells of the pancreas release insulin in response to a meal, in response to high blood sugar. The target for insulin is the liver, muscle, and fat cells. The idea here is your body is saying, oh, I just had a meal. Lucky me. I need to store that glucose into the cells that are good at storing glucose, because I have no idea when I'm going to get another meal. Remember, we evolved in a system where, food wasn't just a matter of going over to Saga. Food was, hunting and gathering food. And you never knew when you were going to get another meal. So we stored it. We stored calories in the liver, in muscle, and in fat. Insulin is important for that storage of food after a meal, especially in a setting where you may not get another meal for 2 or 3 days. The receptor for insulin is the receptor tyrosine kinase class of receptors. It's a tyrosine kinase. So now all of you should be remembering, oh yeah, binding, dimerization, adapter proteins, RAS, MAP kinase cascade. That should be sort of directly coupled in your brain to tyrosine kinase receptors. The effect is to take the glucose up, like I said, and store it. To store it as glycogen. To store it as fat. To store it as protein. Let's not waste it. Let's store it. So this is the story of insulin in a nutshell. Here is the structure of insulin. So this is pre-pro insulin. So what if I know it's pre-pro insulin, I know I'm looking for a signal sequence, and there it is. The signal sequence at the N terminus. Gets cut off, where? In the endoplasmic reticulum, the lumen of the endoplasmic reticulum. So if I cut off the signal peptide from pre-pro insulin, I make pro-insulin. Okay, pro-insulin then makes some disulfide. These are not dollar signs. These are S's. So this is a disulfide. Two necessary disulfides are formed in the Golgi apparatus. There is additional cutting. There's a cutting here and a cutting here. But now these two pieces of peptide are held together by disulfide. And this cutting off of this red part here, it's not red of course, but this loop makes the final product of insulin. So, this is a classic story of pre-pro processing to the pro-hormone processing to the hormone. Here is a slide that I actually love, in a way. So I've given it to you with blanks, and let's tell the story. So this is the story, the mechanism, the molecular mechanism of how glucose in the blood stimulates insulin release. I like this story because it integrates cell physiology and it integrates earlier material from this course. So, heads up, I love this story. I'm just telling you, and so I could ask you a question about it on an exam. Glucose in the blood. You just had a meal. Uniporter. You heard about, glucose transporters in cell phys. So, facilitated diffusion. This is not active transport. This is passive transport of glucose from the blood into the cytoplasm of the pancreatic beta cells. They and other cells express a glucose uniporter, a glucose passive transporter. It's called GLUT2. I will not test you on the name. Okay, what goes in this box? What do all cells do with cytoplasmic glucose? This is a cell phys level question. I want to hear a word, begins with G, ends with S.
Glycolysis?
Glycolysis. Glycolysis goes in this circle. What is the product of glycolysis? This is a cell phys review. I won't test you on this, but I'm trying to get you to remember some cell phys because you shouldn't just throw away all your knowledge from one course and not when you take another course, at least not these courses. What is the product of glycolysis? Let's call two products of glycolysis. ATP and next one begins with a P. PY. Pyruvate. ATP and pyruvate are some of the key products of glycolysis. Okay, ATP, there's ATP. We're going to need that. But obviously this is a cell that has more than just glycolysis going on. What happens to pyruvate? What do we do with pyruvate? I'll give you a hint. We oxidize pyruvate with another cofactor to make a cofactor that begins with A. It actually ends with A too. Acetyl-CoA. Acetyl-CoA feeds into the Krebs cycle. What are some of the products of the Krebs cycle? I'll give you one. GTP, which we can convert to ATP, no problem. What are some of the two other important products of the Krebs cycle? One of them begins with an N and the other begins with an F.
FADH2?
That's the F one.
NADH?
NADH. What do we do with those molecules? What do we call them electron carriers? What do we do with let's just say NADH. What do we do with NADH? We feed it into the three words of the mitochondria of these cells. NADH provides high energy electrons for the electron transport chain. Remember, we convert the energy of electrons into a proton gradient, and then we use that proton gradient to make more ATP. So, I will not test you on cell phys. You've already been tested on cell phys, but I'm trying to emphasize the fact that we're using cell phys now all the time. It's in the background, and now I've just brought it up to the forefront. So what goes in this? So this is too simple. Your slide should have been decorated with more information. The point is, glucose, following glycolysis, oxidation of pyruvate, Krebs cycle, ETC, electron transport chain, ATP synthase, we make a lot of ATP. Okay, so that's the main point. Okay, these cells and other cells make ATP from glucose. Now it gets interesting. And it's not reviewed. Well, it's kind of reviewed from the first part of this course. These cells express a peculiar potassium channel. It's called the ATP blocked, well, it's called the ATP dependent, but it's not dependent. It's an ATP blocked potassium channel. So what happens to these cells when the ATP concentration goes up? Well, we block these potassium channels, but what word goes in this blob here? One word. What happens to all cells if we block a potassium channel in their surface membrane? Pretty much all cells.
Depolarization.
There's the word, depolarization. Blocking potassium channels leads to a depolarization, less negative inside. Okay, now your brain should be off and running. What goes on, now we're going to couple depolarization to release of a hormone. What goes in this slot? In this blob? What essential steps are necessary to couple depolarization to insulin release? Voltage-gated calcium channels. Depolarization opens voltage-gated calcium channels, and now you're off and running with that story that you might be tired of already. Calcium influx binds to synaptotagmin, snare conformational changes, fusion of vesicles, insulin release into the blood. You can sort of understand why I love this story. I hope you can understand why. You may hate it, but I just want to confess that I enjoy it. It integrates so much. So now we've released insulin. Now we're going to talk about the cells that respond to insulin. Fat, muscle, liver. Here are the insulin receptors. This slide I don't love because it sort of black boxes things. But activation of insulin receptors is going to lead to lots of things inside the cell, including changes in gene expression, cell growth, cell differentiation. Let's leave this aside for now. What are the steps between, so here's binding, and here's the effect. What are some of the steps that are missing between binding to these types of receptors and the effects? There's, I don't know, let's see, four steps that I do want you to know. What happens after binding to these types of receptors?
Dimerization?
Okay, we see that. That's not missing. We see dimerization. Then what? Conformational change. Okay, that's always a good conformational change. Sure. And then what? What do we call these? What is this class of receptor?
Tyrosine kinase?
There's the word, receptor tyrosine kinase. What do kinases do?
Phosphorylate stuff.
Right, so what happens after dimerization and conformational change associated with this receptor that now I've given you a hint on, that we use the word kinase in? Or phosphorylation of the receptor. The receptor is a kinase. It phosphorylates itself. Then what? binding of what happens after receptor phosphorylation? Binding of anybody? Adapter proteins, activation of RAS, activation of the MAP kinase cascade, and now we're off and running. So what is missing in this box is cross-phosphorylation of the receptors, binding of scaffolding, or you just write adapter proteins, I've changed the jargon a little bit, adapter proteins, RAS, MAP kinase cascade. Activates those cells, changes their gene expression, increases their expression of a glucose transporter, a glucose passive diffusion transporter, allowing more glucose to come into these cells. Remember, that's the goal of these cells, is to take up glucose and turn it into other things. Those other things are the enzymes necessary to synthesize glycogen, the enzymes necessary to synthesize fat, triglyceride and lipid, the enzymes necessary to convert sugar into protein, into amino acids, and then into protein. So all of the enzymes that are embedded in these words require a lot of enzymes. Where do we get enzymes? Enzymes are protein. They're the products of gene expression. So by changing the gene expression, allowing more glucose in, and rapidly converting it into things that we can store glucose as, these cells, fat, liver, and muscle cells, store the glucose that we got from a meal. Insulin, done. Glucagon is the other hormone that is important for homeostasis of blood glucose. So glucagon is also a peptide hormone. Its response is to low blood glucose. Low blood glucose triggers the alpha cells of the endocrine pancreas to release glucagon. So it's the same organ, the endocrine pancreas, but two different cell types. Beta cells release insulin, and alpha cells release glucagon. The target cells are the same cells that insulin is targeting. The receptor is very different. It's a GS couple. This story, you know this story. You might be sick of it. The effects of glucagon are the opposite of the effects of insulin. Insulin is after a meal and you want to store calories. Glucagon is when you're starving and you want to release those calories into the blood. So glycogenolysis, hydrolysis of glycogen, breakdown of fat, decreased glucose uptake, decreased protein uptake, and decreased protein synthesis. We don't want to be building things now. We want to be making blood glucose. Why do we need blood glucose so much? Our brain, which we need, does not have any storage facilities of its own. Muscle, fat, and liver provide the energy, the glucose, that our brain requires. If our muscle, fat, and liver don't provide the glucose that our brain needs, we're goners because our brain runs out of energy. So we need blood glucose to feed our brain. And that's where this is coming from. Glucagon, between meals, release of stored calories into blood glucose for the brain primarily. So I want to end this with a concept that those of you who have taken AMP2 already know. The concept is homeostasis. And some forms of homeostasis are by the system of